'Cause baby, now we got bad blood. Now we got bad blood. You know we got bad blood. You know we got
Today we'll be talking about the blood coagulation signaling pathway. Early in the semester, we talked about a different aspect of blood, which was the use of hemoglobin, which functions as a tetramer to reversibly bind oxygen in a cooperative manner using this porphyrin ring. Today, we're going to talk about the proteolytic activation of fibrinogen and its conversion into an insoluble form called fibrin. This is related to quite literally patching holes in blood vessels to prevent an organism from bleeding out. It is a highly regulated process, and I wanted to spend some time explaining this to you because it's directly related to what you were doing in the lab portion of biochemistry in terms of purifying things from rabbit plasma. What is actually in blood? For some of you, this might be review, but for others of you who don't have a biology background, I wanted to go over this briefly. Basically, you have cells shown here, and the stuff that the cells are moving in, the liquid. The cell types found in blood and in the ending sites, and you have erythrocytes, leukocytes, and thrombocytes, which stand for the red blood cells, white blood cells, and platelets, respectively. On Friday, I'll talk a little bit about platelets. For the most part, we're going to ignore these things. The focus of today is the plasma component, which includes essentially the liquid part of blood, but also all of the soluble proteins that are in it. During the process of coagulation, some of these proteins become insoluble, and that's what generates the gel-like substance or the hard substance of a clot that prevents you from bleeding. There are links in the original PowerPoint if you're interested in learning more about these components. So on Monday, I asked a question towards the end of the lecture, which was trying to give you just a biological feel for what this looks like. A lot of the times in biochemistry, you don't see what you're working with, by visible light anyways, because you're dealing with soluble proteins. But in this case, you can actually see the protein directly because it's forming these long strands. So the stuff in light pink was this mesh-like network that was trapping certain cells within the blood, like red blood cells or the platelets. And this is, you're forming a blood clot. So fibrinogen, which was the structure I showed on the first slide, was the structure of what would be soluble fibrin. But there's a process by which, excuse me, this is in the way, in which this soluble form of the protein is converted into an insoluble form, and it must be tightly regulated. If I were to focus on a figure, in this chapter, this would be it. Basically, I want you to understand how this pathway is regulated both positively and negatively. If you look at your figure legend, so this is actually figure 6-41 in your textbook, the coagulation cascade. The point I'm trying to make here is that there's lots of other soluble proteins and enzymes in plasma, not just fibrinogen. So do you see where fibrinogen is on this pathway? Right at the very end. This is the soluble form of the protein, and it's going through a cleavage event to become fibrin, and then it starts to aggregate. And there are multiple steps that we'll go through in detail. I want you to understand in detail what's happening in this pathway. In lab, when you did your first ammonium sulfate precipitation, you were hopefully isolating a fraction of proteins that was enriched for fibrinogen. The reason for this is that proteins precipitate at different, they have different propensities to precipitate. Since blood clotting requires the precipitation of proteins, it makes sense that the components of the blood clotting pathway, like fibrinogen, would come out first, right? They're primed to precipitate toward after getting some sort of signal. In this pathway, the end is down here dealing with just fibrinogen or its cleavage into fibrin, but it starts with vascular injury up here, right? And then essentially what you have going on here is a proteolytic activation of zymogens. Zymogens are inactive precursors of enzymes such as proteases. Thrombin is the most predominant protease in this pathway. It does all sorts of things both early in the pathway as well as late, including direct cleavage of fibrinogen into fibrin. But you also have a slew of other proteases. In this figure, all of the proteases are shown in blue. And it gets a little confusing because they use Roman numerals, but we'll go over that. But you also have, in addition to these proteases that are in Roman numerals, you have things like protein C. Protein C is the only other protease other than thrombin. And you can see when you have a zymogen, here, let's focus on protease 10 or X. So which form is the zymogen? Here. So this is before this protease is cleaved, and after it's cleaved, it's activated. So this is protease 10 activated. It's sort of this dizzying phenomenon, I guess you could call it, whereby you have cleavage of one protease making it active, which then cleaves another protease making it active, which cleaves another protease making that active, so on and so forth. The big message, the big take home I want you to have from spending a little time studying this particular pathway is that there's both positive and negative regulation. And that's really important because it makes sense to form a clot, right? On one hand, you want to be rapidly activated in response to some damage that could cause the loss of blood, but you also want to turn the pathway off once you have formed some sort of clot. Otherwise, you have the opposite problem of now preventing blood flow and you can die by the other way, right? You have blockages within blood vessels. The inhibition of this pathway is indicated in these red X's here. AT3 stands for antithrombin 3. Thrombin is, I believe, a serine protease. And all of these other blue proteases, I believe, are related to thrombin. And that is why an antithrombin 3 inhibitor, can also block these other proteases, the activated forms. Okay. These are a little bit different, and I'll cover on Friday, than AT3. You can also see there's a metal cofactor that's really important for some of these proteolytic steps, and that metal ion is calcium. So it's involved in the catalytic mechanism of the proteases. If we take like just a step back, I guess, why did we do purifications of rabbit plasma? Why are we so interested in blood coagulation? From a patient perspective, it doesn't need much explanation. But from a historical perspective of how sort of the field of biochemistry came to be, without oversimplifying it too much, in the early days, it was biochemistry was very intimately related with the field of hematology, the study of blood. And so patient care is one reason why we're studying this pathway, but a good rooting in scientific history, in science history, is a second reason, and then the third reason is somewhat, albeit it's just practical. The previous biochemistry professor at Augsburg did a lot of research and has a ton of rabbit plasma just sitting in the freezer. So it's a good sort of introduction to protein purification. I'm a little bit sad due to the circumstances that we won't be able to continue with lab. One of the things I'd hoped to do with you was to purify just components, working with components that were just involved in blood coagulation and doing a little blood clotting mini assay. One of the things we are interested in doing research is the effects of other metal ions, not just calcium, but things like zinc, mercury, other compounds in the environment that might impact blood clotting. It's an answer that is not really known and the subject of ongoing research in my lab. This is complicated, but it's actually in the textbook. It's a lot more complicated in reality, even compared to the textbook. There's tons more enzymes that are involved. This isn't even dealing with components of the immunological response pathway, which is just not gonna go there. But if you do want to go there, you can click here in the PowerPoint. Merck is a biotech company, and they seem to have a lot of really nice pictures when it comes to this pathway. But as you can see here, there's all sorts of other things going on. I'm not going to go into detail other than this is just kind of like the wow factor, I guess. A reminder that this is a prelude really to the chapter in your textbook that focuses exclusively on signaling cascades, but I'm focusing actually on chapter 6, which is more about, it was talking about enzyme regulation at the tail end of it because it was related to lab. But This is a preview of where we're headed Friday and next week. It's kind of like telling a children's bedtime story. Like if you think of most of the Disney movies, especially the older ones, they're all basically sort of like variations of the same story. And in a lot of ways, that's true for signaling cascades. And that's how I'm choosing to try and present at least some of this information to you today. But the main components of the story are there's a signal, some sort of conflict that needs to be resolved. In the context of blood coagulation, the signal is, of course, tissue damage. So this is the signal, the physiological signal that needs to be translated into some sort of cell response. And my goal is to give you some good rigorous experience, because you've covered signaling pathways before. Well, most of you have. But here I really want to actually give you, not just talk about it at the surface, but some actual good experience talking about the regulatory how the regulatory enzymes are modulated. What I mean by this is there are enzymes in this pathway that regulate the response to that signal. In truth, a lot of you already in your protein presentations got at this to an extent. In fact, Faith and Cal talked about, I believe, pepsin, which is another example of a proteolytic. It's a zymogen that gets converted into a protease. So there's the signal, some regulatory enzymes. You can see there's some Roman numerals here, 2A, and then there's thrombin. So if we were to zoom in on one of these regulatory enzymes, the idea is that at the biochemical level, at each of these points, there's sort of like stoplights, right? And there's a series of stoplights that goes on. And you can either inhibit or stimulate this regulatory enzyme's activity. What were some ways to regulate enzyme activity? Right? How do you start changing the status of whether an enzyme is active or not in a biological setting? Post-translational modifications, right? You can start adding things to them. We talked about this on Monday, but I just wanted to show you a picture showing you the range of post-translational modification sizes. This is the phosphate group. This isn't even recording. I really hope it's recording on my computer. This is the third time I'm doing this. So that's why I appear a little tired. Excuse me. So phosphate is 100 Daltons. Farnesyl group is 400 Daltons. And then, as I said Monday, you can even add entire proteins. So for context and size, is basically a massive thing that you're appending onto your regulatory enzyme. And here's the size of the little phosphate group. So, well, what you're doing when you're adding these things is a number of things. You can be directly changing the enzyme's activity by causing a conformational change. You can also be impacting the localization of your regulatory enzyme. This is true for the protein that is implicated in progeria. For those of you who have started watching the Life According to Sam documentary and reading the associated literature, you hopefully know that the protein involved there is lamin A. And lamin A is farnesylated. So this is added to it. And this kind of looks like a lipid, right? It basically causes your protein to be recruited to membranes. And lamin A is it's mostly in the nucleus. And so it causes proteins to be localized to the inner membrane of the nucleus. I'll talk about that a little in a different presentation as it's related to progeria. But I just wanted to mention that since it's something related to the last assignment for this course. So in each of these cases, you can either add or remove. It's not like every time you add a phosphate, you're always activating the enzyme's activity. It really depends. Sometimes addition of a phosphate can inhibit its activity, right? And there are dedicated enzymes that both add and remove, add or remove these post-translational modifications. Tina and Jennifer talked about Src kinase. A kinase is an enzyme that adds these phosphate groups. A phosphatase is something that removes these groups. Anyways, there's, so this is one broad class of post-translational modifications in the sense that you're adding a new thing to your regulatory enzyme or removing something from your regulatory enzyme, these chemical groups. In a second sense, the second type of post-translational modification is to remove a piece of the enzyme, of your regulatory enzyme itself. So in this case, you're just removing a covalent bond and you're actually taking out a piece of your protein. I mentioned that we had a nice talk already in the class that discussed this briefly, but your textbook also goes into it in more detail. So these are all of your, these are the proteases that digest other proteins. They're secreted from cells and they're in your digestive, like various parts in your body that help you digest protein. As you can imagine, just unregulated digestion of protein would be disastrous for a cell. And so that's why potentially there's this, I would say, more regulated form, right? In the sense that you really want to be careful. So you have to start cutting up this protein first. You make sure it's inactive until it's absolutely needed. A zymogen, sometimes it's called, you'll see like a pro in front of it. Like pro, what is the enzyme we're focusing on today? I have to actually look back myself. Pepsin or thrombin. So here, for example, you see prothrombin. Pro is often used to designate a zymogen form. Other times it's just the addition of an A to indicate the proteolytically activated form, and the form without the A would be the zymogen. You can have endo or exopeptidases. So these are the enzymes that are cleaving your zymogens to make the active form. Here we're showing chymotrypsinogen is now chymotrypsin, and it was cleaved by trypsin. It was cleaved at the very end of it, right? So it's this would be an exopeptidase. But you can also have internal cleavages. Well, don't worry about endo versus exopeptidases. The point is that there are many different cleavages that you can make. Okay, so I thought I'd talk to you a little bit about fibrinogen. What do you see when you look at this ink blot? If you're like me, maybe you'll see fibrinogen and know then that you are ready to graduate and be done with this craziness, right? You're ready for the real world. Get the hell out of here. I'll miss you. But sorry, fibrinogen is a what in terms of quaternary structure? What do you see when you look at this? It looks like spaghetti, just woven in around itself. But how would you describe its quaternary structure in biochemical terms? Well, you see that there's alpha, beta, and gamma, right? So there's three, and then the alpha, the green parts have this knob, that's just like a domain. But these alpha, beta, and gamma are different chains. So they're different peptides, right? They're not covalently bound to each other. They're actually different proteins that are associating with each other at the 4-degree sign quaternary level. And this is similarly just a specific domain within the beta chain. So there are 3 chains that are associating with each other, right? But actually, if we were to follow it, this beta knob right here, let's follow the noodle, belongs to actually this part over here, beta. So this chain of 3 peptides is linked with another chain of 3 peptides. So maybe try and think using some words like monomer, dimer, trimer. Describe what you see. What would you say this protein is? Well, we certainly have a trimer, thinking of the 3 musketeers. That's one part of it. And actually, I just told you that this beta knob belongs to this other one, but ignore that. I'm oversimplifying this. So it's a trimer. Is the trimer the same or different peptides? Different. So it's a homo or heterotrimer. It's a heterotrimer, right? Heterotrimer would be the first word because trimer means 3. And then it's 2 of those. So it's a dimer of a heterotrimer. Okay. Fibrinogen versus fibrin. Fibrin refers to when we cleave off 2 parts of, I believe it's beta and gamma. You'll double check me. You cleave off 2 little parts of fibrinogen, and now it is fibrin. Okay, so there's the protease that cuts it off is thrombin. So thrombin cleaves off. There are 2 tiny fragments that come off of fibrinogen, and once you make these cleavages, it can start to aggregate. Fibrin simply refers to the cleaved part that is prone to aggregation. So where are we in this pathway? Let's start at the end, okay? I just want to walk you through some of the major, it's a fine, whenever you're talking about signaling cascades, it's a fine line between, at least to me, being busy work, but it's not just busy work, because that's exactly what signaling pathways are all about, is not just calling it busy work, but saying, no, we care about each of these steps. So it's a fine line between busy work and seeing the overall themes. So I hope to walk that line with you today. To help me get through this chapter, I had to use an analogy, which is why I made that this week's activity, because otherwise my brain just melts when I look at this and I start talking about, for example, 10 gets cleaved to 10A, which cleaves 7 to 7A plus a tissue factor And the 7A plus the tissue factor can then reactivate 10 into 10A. Aside, it's just like, what the hell is actually happening here? Like, right? So the analogy I came up with was, is it Snow White and the Seven Dwarfs or Cinderella and the Seven Dwarfs? I keep getting them mixed up. Snow White, I think it's Snow White and the Seven Dwarfs. But then you have Sleeping Beauty, and I just get confused. So some princess who's going to be sleeping, who needs to be woken up, is my story. I think I'm making a hybrid Disney story at this point. But the idea here is that the Seven Dwarfs, there's not actually 7 proteases, but there are a bunch of proteases. Okay? So whether it's helpful to you or not, that's what I'm going to do. But I'm doing it with the intent of being helpful. So let's start at the end. And here's our princess, or I don't know who I have shown here. We're going to assume this is we'll call the inactive form in terms of forming a clot. And throughout this story, if you look really closely, they have lost their feet. So there's been a little cleavage event here. That's why it's now fibrin, not fibrinogen. And then there's going to be some polymerization event. Now we have a soft clot. And now, soft clots are good, but not quite good enough. So we want to make a hard clot. So we're going to actually covalently cross-link this. Let's start here. Snow White and the Seven Dwarfs. No. Today, once upon a time, there was fibrin, or fibrinogen, who was put to sleep and needed to be woken up by a series of proteases. One of these proteases was really important. His name, or her, I really don't care, was Prince Charming, or Prince Florian. I don't know. Anyways, the point is that there's a signal, and we have multiple characters in our storyline that have to activate our Snow White, or Sleeping Beauty, or Cinderella, whoever the heck it is. So step 1, actually the end, this is the last step. I'm going to work backwards with you. Is that fibrinogen gets converted to fibrin. This would be, you know what, let me pause this really quick, because we should know which parts get cleaved off. It's actually pretty important. You know what, don't worry about it. It's something that I'm interested in, but maybe you don't have to worry about it. Here I'm showing you thrombin, not prothrombin, because Prince Charming is missing his body. He is not a zymogen. Remember, all of these proteases, to be active, have to be cleaved. Once it's cleaved, once fibrinogen has been converted into fibrin, I should say fibrin, now we have, oh, I thought that was kind of cute because it kind of looked kind of like that. But anyways, this keeps going both across and this and up and down. That's what this structure is that's shown in the textbook. This is important, right? Because it's making a really compact and there's lots of interactions that are strengthening this mesh work, because you want to be able to plug a hole. So we're focusing at the end part of the pathway. We're starting to aggregate in many different ways. In one of the fairy tales, there's the princess likes this bluebird. So let's make one more analogy here. which is that simply aggregating it in and of itself is not enough. We need to form covalent bonds between parts of the structure. So this is the first, this is another type of post-translational modification where a covalent bond is formed. It's kind of like a disulfide bond, except instead of being between two residues with sulfur groups, it's between a lysine and glutamine. So there's a transglutaminase at the end of this pathway called 8. And what you should know about 8, it's not a protease. It is a transglutaminase. It must be cleaved, right? And now it's active. So that, if there's one big theme to this whole thing, it's that it's just everything's getting cleaved. And in the uncleaved state, it's inactive. When 8 is active, 8A, it can form a hard clot. So it's kind of cool. If you take fibrinogen, you add in thrombin in a test tube, and you let it incubate for a while, and then you try and like turn the tube, you'll see it's starting to form a gel-like substance, but it's not really hard. At some point it'll become like a gel. But in terms of stopping bleeding, right? If you think in the context of your circulatory system, there's a lot of pressure, right? So the clot not only has to be gel-like, sometimes it actually has to be like physically hard, able to bear a high force. That's what little bluebird or 8A does by making these covalent bonds between, it's not an intramolecular bond, so it's not within fibrin, a single fibrin molecule, but between fibrin molecules within the context of this big, crazy structure that's forming. Where were we in the pathway? I'm just bringing you back to the figure in the book. We're down here. I just told you the end of the story. I blew the ending. Spoiler alert. I told you the ending, how the story ends, but it's okay. Okay. Hi ho, hi ho, hi ho. It's off to work we go. I think that's how it goes. And then they whistle, but I can't. So now let's focus on the earliest parts of the pathway. Okay. I think it's worth noting that there are often, okay, let's keep track of the themes in our movie, right? Theme 1, everything gets cleaved. It's like you meet a character, it's like, hey, they're not cleaved. I wonder what's gonna happen. They're gonna get cleaved. The other theme of our movie is that there's 2 arms to our pathway. There's an intrinsic and an extrinsic pathway. I'll focus first on the extrinsic pathway because the extrinsic pathway is what is responsive to the signal, how our movie began, right? In this case, the signal being vascular injury. But let's talk about some of these 7 dwarfs. They're cleaving each other, and they're making feedback loops. So that's the third theme. Theme 1, maybe I'll write it down for you. Theme 1, themes, cleavage. We have cleavage, dreams. Oh God, dreams. You know what, you guys, I'm sorry. And I'm not going to be able to go back and edit this. I tried editing the previous 2 versions and the program kept crashing. So you'll have to I tried to edit out my mind wanderings. Sorry, even online, it's unavoidable. So I was trying to think, I really want to have kids. I'm sick of career. And you shouldn't say that out loud, but who cares? You know, social distancing is making me a little crazy. So I'm pretending like I'm explaining this to my little kid that's just been born. Um, and I would tell them that, well, there were these 7 dwarves, right? Um, some of them were happy, some of them were not. Um, the ones that were happy were easier to activate. So they're actually going to be involved in the first part of the story. Once that's the extrinsic part of the pathway. The first thing that will be, the first protease that will be involved in the story is Dopey, and Dopey will activate Doc. Doc is just really smart. He doesn't excel in being organized in his thoughts or, you know, being clear in general, but he's good. The thing that he does really well is that he activates Prince Charming or Thrombin. And that once you activate Thrombin, the story is really starts to get interesting. So this is going to be the extrinsic pathway, Dopey and Doc, or proteases 7 and 10. And then the second part of the story will be how we activate Grumpy and Sleepy. Grumpy being 11, Sleepy being 9. There are actually 2 more dwarfs that I'll talk about, but I didn't have time to make a slide including them as well. But hopefully you appreciate that I was trying to make the proteases involved in the extrinsic pathway, those that had positive emotions, and those in the intrinsic have like either negative or just neutral emotions. And Sleepy is one of the later proteins to act in this pathway, which made sense to me. Functionally, what are the differences between these different arms of the pathway? The extrinsic pathway does initially lead to a burst of activated thrombin. And so you can get clots that form, even hard clots if the transglutaminase is activated. But it's not an all or none thing. You have an initial burst of thrombin, but you need to sustain those levels of activated thrombin, or else the clot will start to fall apart, and it certainly won't grow. So let's talk about the initial burst. Excuse me. I really miss talking to you guys and meeting with you in person. I just want to say that, like, it's really hard to teach in this manner because then I'm just like talking to myself. Have you ever seen the movie Moon? Moon, Moon. It's a sci-fi movie. You should watch it. I feel like the character in Moon right now. Okay, let's go back to our pathway. And the question I posed to you, here we are, I showed you Dopey. I want to focus on protease 7. VII means 7, yes. So the question is, what cleaves Dopey? Pull it up and actually look at it. The answer is 10A. Okay, that's what this figure is showing you. You had a blue protease cleaving 7. This is the zymogen into the active form 7A. Good job. What cleaves doc? What cleaves this into this? 10 into 10A. Discuss with your pet or child. Well, the answer wasn't so simple this time, was it? Well, we have one of the answers. You can see there's 3 things pointing to this conversion of the zymogenic form to the proteolytically active form. 7A is one answer, dopey. And we'll get this to later parts in the story, but another answer was IXA is 9A. We also have 8A, right? So it's interesting. This one protease can be cleaved. It can be the cleavage of this one thing is activated by many parts of the pathway, right? Are you familiar with the idea of like the issue of which came first, the chicken or the egg? So here's the issue. Maybe you're seeing, right? 7A is needed to cleave X into XA, right? But how does 7A get cleaved? XA. Okay, but then how does XA get cleaved? Because you see what I'm saying? So here's the thing. How about you think about this, answering this question basally, without any vascular injury. This pathway needs to be turned on or off. Off. You don't want blood clots forming, generally speaking. So which form of the proteases should they be in? Cleaved or not? Cleaved or the zymogen? The answer should be zymogen, right? Otherwise, you would have different parts of the pathway activated, ultimately leading to your princess having her toes or feet chopped off and then aggregating. Good job. So, and that is by and large true. In the absence of vascular damage, all of these proteases are in the zymogen form, but that creates a problem because you need at least someone to be initially activated, right? It's the vascular injury in and of itself. Yeah. So, yeah. So maybe how would you fix this problem? If 7A is needed to cleave 10, if 10A is needed to cleave 7, and both start out as zymogens, how could we possibly solve that problem? Come on, dig deep. Have you ever heard of autocatalysis? So it turns out that 7A, dopey, because it's just a little dopey, for example, it actually, a very small percentage of this particular protease in the blood, right, in the blood is actually already cleaved. So, but then that leads to another problem, doesn't it? If you even have a small amount of 7A, right? Wouldn't that cause there to be cleavage of 10 to 10A, which would make more 7A? And then you'd have, oh no, and then all of a sudden you have a blood clot? Absolutely, good point. So how would you solve that? There's two answers. I'm going through the logic of signaling cascades with you. There are two answers. The first is these red X's, right? These red X's in part ensure that it's not like a chain reaction leading to a nuclear explosion, right? You can turn on the pathway. You can have some initial vascular injury, which makes maybe a little bit more cleaved protease. But in the absence of sustained signal, these inhibitors, AT3, and also protein C and protein S, will turn different branches of the pathway off. Okay, so simply having a tiny bit of 7A isn't actually going to turn the whole pathway on. There's actually a second answer, though. What does TF stand for? Not transcription factor. We're not talking about gene regulation. But we are talking about tissue damage. It is a tissue factor. Tissue factor is actually located in tissue, such as your blood vessel, like the blood vessels. You can see here, in order to convert 10 into 10A to make Doc happy, and who then activates Prince Charming, right? In order for this to occur, dopey is not enough. Dopey needs a little help. Dopey needs tissue factor. Where are we? Like, where are we in an actual cell? I'm going to actually stop this video at this point. I'm going to keep talking, but then try and edit this in and show you this. But I'm just going to say it in words right now. Where are we in the context of not just a test tube, but the body? All of these factors are located on the inside of a blood vessel, right? Where your blood is located. Correct. All except one. Which one is not located in the blood vessel? Tissue factor. It is in the tissue. So 7A, this first protease that gets things started, is physically separated from transcription factor by a lipid bilayer. If you break the lipid bilayer, which is vascular injury, now tissue factor can help 7A convert your first protease into the active form, which now you really get things rolling. So really the story isn't so much about 7A, the first part of the story. It's like a love story. It's the breaking of barriers. Let's tear down those walls, okay? Because these two, they love each other. They really do. They need to be together in order to function. And that's what happens. Okay? So the key here is we brought up a number of interesting dilemmas, even in the first part of the movie. One of them was that neither 7A or neither of these proteases would ever be cleaved. Perhaps this first protease has some autolytic activity, autolysing of itself. Yeah, there's a small amount of it present, but it's not sufficient to just turn the whole thing on, because we also have inhibitors of these proteases, plus this first, even if you have a small amount of 7A present in the plasma, it can't function without TF, tissue factor. So it truly will be in response to some sort of biological event. Again, it's easy. That's the challenge of biochemistry. It's like you have to span both worlds, and it's tough. We're just lost souls, you know. Some of us are more chemists than bio. Some of us are more bio than chemist. I'm more bio than chemist, which is probably why, because a lot of you are more chemistry. Anyways, I try. We all try. And you've done a great job too. And I need to move on. What is this? Oh, will it show it? Let's see if it does it. Play. Oh, it's gonna
Okay. So what I have diagrammed for you here is 3 different cells. Okay. These, it would extend further in both directions, but this represents the outer lining of a blood vessel. Okay. And on the inside, down here, this is where your blood and plasma is located. So if I restate the initial question, it was we had 2 things we were looking at. 7, the zymogen form having to be converted to 7A to be active.
That's dopey.
And 10, the zymogen form needing to be converted to 10A, the active form. In order to get 10A, you needed 7A and vice versa. So
sure did.
Part of the first part of the solution to this problem at the biochemical level, okay, was that there's always a little bit of this present. There's always a little bit of 7A present. But then the question you should have been asking yourself is, well, if some 7A was always present, then you'd always have cleavage of 10 to 10A, and then you would go down the pathway and always have a clot forming. This is where the second part comes into play of TF, the tissue factor. It's called tissue factor because it's located in the tissue itself. Okay, so here, let me use a new color. Thank you, Navi, for mentioning this app. It's called Notability, and so I'm trying to use it.
Thank you, Navi.
So TF is here. It's in the tissue, part of the blood vessel, right? The point is that it is sequestered,
separated by
unable
a
to
lipid
interact
membrane.
With 7A. And remember, you need TF for 7A to work.
Sure
So
did.
simply having a little bit of 7A present is not actually going to cause you to go down this route. This is where the biological signal is incorporated, where it becomes important. And what is the biological signal? What do we want blood clotting to occur in response to? Damage of tissue, a break in the cell wall. Okay? So it's a combination of having some 7A present, but also this break, an actual damaged tissue. This is in part what allows the first part of the pathway to proceed.
All right.
Okay.
Yes. Well done, Jason. I'm just gonna double check that if this isn't recording, I'm gonna lose my shit. Excuse my. I do need to be more professional, but it's sort of who I am. It better be recording. It is good. So that was Dopey and Doc. The story's a little interesting, but then the evil, the bad person, the witch or the whatever, comes in and says, no, dopey doc, we know what you're trying to do. You're trying to make a clot. And so this is where the conflict of the movie comes in. The initial conflict was there was a membrane separating tissue factor from 7A, but the secondary conflict is these inhibitors, right? We need more, right? I also want to mention you should really look at this pathway, really look at it. If you looked at it, you might have said, Dr. T, you talked about antithrombin 3, yeah. You also talked about protein C and protein S as being inhibitors, yeah. It's interesting that one of these inhibitors is itself a protease. How does antithrombin 3 actually work? Maybe it's binding allosterically, whereas this is actually cleaving something. It's really interesting. We'll have to come back to this one. I want to point out that there was 2 inhibitors in the first part of our movie. One was the antithrombin 3 that we talked about. The other was this one that I haven't even said anything about yet, which is TFPI. TFPI's tissue factor pathway inhibitor. Very creative. Tissue factor pathway inhibitor. The point here is that in the first couple scenes in our movie, there were 2 different inhibitors, right? Functioning. So let's move on. We're done with Dopey and Doc for now. Although that's the thing about signaling pathways, you're never really done with any part of the story because all parts of the story are related. Hey, you see what I did there? Hey. So the extrinsic pathway is not sufficient for a sustained clot, although we did have an initial burst. This in and of itself, if there was a little bit of vascular injury, Dopey and Doc, they got their stuff together a little bit. You can see from activated 10A, you do come down here. Oh, yes, you do. You do come down here and you do make some thrombin, okay? So there is actually a little bit of blood clotting forming, but for a sustained clot, nah. Why? This is why I talked about TFPI, tissue factor pathway inhibitor. This one's really good at shutting down the 7A tissue factor complex, right? So again, it's another level of regulation. It's layers and layers of regulation like you would not believe, okay? The point here is that even if you have a tear in the blood vessel and the initial love story, oh, they found each other. Well, even though they found each other, guess what? TFPI is coming in and saying, get out of here with that romance. What do you think this is? This is a horror movie. This is, get out of here. This isn't a love story. You already know that. Red dash stuff means inhibiting. Seriously though, I would like you to be able to, sometimes I hear myself sounding like I'm nagging, and it just makes me feel like, what have I become? The sustained clot requires the intrinsic pathway, okay? This basically, you need to have some sort of feedback loop maintained, okay? You need to amplify the signal. That's why you get other proteases involved. That's why there are not 2 Snow White and the 2 dwarves. No, you really did need the other 5 or so dwarves. This requires the intrinsic pathway, okay? The big difference. What is the big difference between the extrinsic and intrinsic pathway in terms of where they're located, where the components of the pathways are located? The intrinsic pathway, everything is found in the blood. Everything. It's already there. Whereas the extrinsic, one thing came from outside tissue factor. The sustained clot requires this intrinsic pathway. So let's briefly go through the arrows. Let's get lost in the woods just a little bit And let's hope that we come out the other side and that this records. So here we are. Let's focus on the early part of the intrinsic pathway. And here we go. So now we're talking about grumpy and sleepy. Roman numerals, what am I talking about here? X plus 1 is 11. So we got 11. This is a protease. 11A and 9. IX is 9. 9A. It's interesting that one of them doesn't require calcium. I don't know. All components of the intrinsic are in blood. Sorry, I didn't correlate that with what I was saying. So this second feedback loop, you can see, consists of, it's not just the intrinsic, it's the intrinsic feeding back into the extrinsic, which then feeds back into the intrinsic. So here we got dopey, 7A, activating what? 11A, grumpy, activating 9A, sleepy, activating what? Doc, activating more dopey. So like the reason why I'm going through this with you is I might ask you on the final to explain in words what is actually happening, right? So example, answer. Well, I won't do it because it was just what I just said, except a little more focused. So in the intrinsic pathway, the primary, we had 2 inhibitors, right? Antithrombin 3 and tissue factor pathway inhibitor. In the earlier part of the intrinsic pathway, we're dealing primarily with antithrombin 3. Okay. The theme, almost every step in this pathway is subject to positive and negative regulation. Okay. So it's as though there are actually not just one, it's not just one traffic stoplight. There are 2 traffic stoplights, right? Things are simultaneously subject to both inhibition and stimulation. You might notice that this second feedback loop isn't just the 7 dwarfs, but also Prince Charming is acting up here. I don't know if that's the next slide. It sure is. Look at that. So thrombin was also able to cleave grumpy. So there are 2 things that can cleave grumpy. All right. Thrombin also plays roles in the earlier parts of the intrinsic pathway. So here I'm pointing them out to you. Here I just said that thrombin helps cleave protease 11 to 11A. Look, it's also involved here though. Let's call this one bashful. It's cleaving 8 to 8A. What else? Sneezy, maybe. 5 to 5A. In terms of where we are in the movie right now, what is the big theme? Of the 7 dwarves, who are we ending with? Doc. They really all kind of converge on Doc, right? In order to get this, I'll talk about this in a second. This is really important. They're all sort of flowing through these feedback loops. Either here's a feedback loop, right? Here's another feedback. Oh, here's another feedback loop. And then we have thrombin itself, not really feedback loop, but the big picture, the big theme that we're coming across is that 10 is really important, okay? All of these 7 proteases, not 7, all of these other proteases are converging on 10, zymogen to 10A. So you start with dopey, need help of the other intrinsic dwarfs, but really it's only 10A that can actively make more thrombin or prints. So thrombin, right? That thrombin is the thing that ultimately leads to the functional output of making a clot, right? but there's only one 7-dwarf. There's only one protease within this pathway that can convert prothrombin into thrombin, and that is DOC. That's why DOC is really important. So since thrombin does multiple things, or your prints in the analogy, once you make thrombin, it really has an additive effect that is sustained. Because not only is thrombin making the fibrinogen turn into fibrin and then subsequently make a clot, thrombin is also cleaving the proteases that make more thrombin. Where I'd like to go after I have time to read a little bit more is talk about these two aspects of the intrinsic pathway involving inhibition by protein C and protein S. I encourage you to read that in the textbook. A lot of you will deal with patients, may even take blood samples. I remember, Kyle, you were telling me that you were shadowing someone or doing surgery, or maybe it's some part of your job. But when you do surgery, right, you have to administer some sort of medicine to either prevent clots from forming during the procedure, or promote maybe the clots, of promoting coagulation after the operation is over. I'm not sure. I want to show you, now that we've spent this time talking about this pathway, where some common drugs impact the pathway, such as warfarin, heparins, and aspirin. Okay. This is not in the textbook, but I might mention it on Friday. Really interesting. I was reading a little bit about blood coagulation, and I came across this article from the 1930s. So right when they were starting to elucidate this coagulation pathway, there was this weird observation that snake venom promoted coagulation, I believe. So I might try and throw in something interesting about snakes, of all things. I don't know where it targets the pathway, but it's really interesting. So what did we do today? Together, we told a story. We began with this notion of regulating enzymes through post-translational modifications. These post-translational modifications were a metaphor. Excuse me, the traffic light was a metaphor for these post-translational modifications, right? Initially, all semester long, we've studied like one enzyme, and usually we've talked about just inhibition of that enzyme. And even if we've also talked about stimulation, it's always been one at a time. You're either inhibited, something's actively inhibiting it, or something's actively stimulating it. This is where we started today. But as we told our beautiful story together, we ended here. In London, UK, the traffic light tree. It is a piece of art that's not functional, pretty cool to look at. But I think this is really the metaphor that to think of. Simultaneous regulation of even individual regulatory enzymes by multiple inputs. It is complicated. Enzymes are inhibited and stimulated in the context of biosignaling. And oftentimes it's simultaneous. The simultaneity, there I just made a word up, of these pathways is really important. It's not just because biochemists like to be complicated and like droning on forever and ever and ever. We didn't make the blood coagulation pathway. We are simply studying what nature evolved. You know, nature made this pathway overlapping, redundant, filled with feedback loops. This person cleaving that person, but wait, I can't cleave you because I need my tissue factor, all of these different things, right? The reason for this is you need a biological process to be dynamic. We are no longer just talking about regulatory enzymes. We are talking about regulatory enzymes in the context of a signal. So the signal that we tried to focus on today was tissue damage. By being constantly subjected to both positive and negative regulation, it allows the pathway to be very dynamic and respond. Right? It's not an all or nothing thing. And when it does respond, it can respond to different things, right? We just focused on one thing. Oftentimes in pathways, it's like, I'm going to see this aspect of my environment, the cell is saying. And then it's also saying, well, but I also want to see that part of my environment because I need to make a really informed decision. That's what the cell is doing. The last thing I want to say is to encourage you to read pages 230 through 234. Leah, you mentioned that maybe the pages don't correspond directly. So I have posted this on the syllabus. You're right, though. At some points in lectures, I start talking about pages. I'll try and do a better job of saying sections. So page 230 is the section that says some enzymes and other proteins are regulated by proteolytic cleavage and enzyme precursors. I'm asking you to read that. And the next section, which is a cascade of proteolytically activated zymogens leads to blood coagulation. And the thing to focus on is that the figures, especially in this section, including not only the quaternary structure of fibrin/fibrinogen, but also the pathway itself. A few other things, why this is important to you. I wanted to say a quick thing about drug interactions. So if someone were to ask you, now given what you have learned in terms of signaling cascades and how enzymes are regulated, why would this, the question is, do you have a better appreciation for why it's so hard to develop drugs? I hope you do. For example, let's say you want to impact a drug that is promoting or preventing blood clot formation. Let's say you target one part of the pathway. The issue is, even if you target one part of the pathway, it can find other ways to get around that, right? Because signaling cascades are so complicated. I'm trying to give all of you here in this point some comfortability in navigating a signaling pathway in general. A lot of you will end up working maybe in a clinical setting, and you might, when you start working on maybe a certain type of patient with a certain type of disorder or disease, you might be interested, or you might get let's say, be offered by a pharmaceutical rep the opportunity to use a new drug, and you'll need to know what are the off-target effects of this drug. It would be helpful for you to be able to pull up a signaling pathway and see, where am I impacting the pathway? And then overall, this is why treating disease is difficult. So with that, the story ends, at least for today.